Okay, so last class we talked about malaria, we talked about AIDS. So we'll cover TB. We talk about measles and smallpox. We talk about antibiotics and antibiotic resistance in this class. We'll do some questions to test our understanding, and this will be the last video for infectious diseases. And I'll follow up with chapter 11, which is immunity. So continuing with TB. So TB is caused by Mycobacterium tuberculosis and Mycobacterium bovis. So the type of organism is bacterium. So the transmission is by airborne droplet or droplet transmission. This is for Mycobacterium tuberculosis and also on the cookie on pasture rice milk, and this is for M. bovis. So what happens when a person inhales TB or the TB particles by droplet transmission? So the bacteria will enter his respiratory system and then travel along it and reach the alveoli. And over there, phagocytes will firstly engulf it, but then the TB itself will survive. Eventually, more and more macrophages will arise due to chemotaxis. So chemotaxis, we'll talk about that in chapter 11, but chemotaxis here, this simply means that chemical substances are attracting white blood cells towards it. So macrophages will eventually surround the bacteria and engulf them, and then they will eventually die and release the bacteria. So this falls off. This basically results in the formation of a wall of adhesion, and this forms a tubercle. So when a tubercle forms, the bacteria will divide within it by binary fission. Eventually, they will burst, damage the alveolar bronchial wall, and then again it will start spreading. So when the tubercle forms, we say that the TB, it will last within the body for a long time. There's also antigen concealment, as we talked about that in malaria, when the white blood cells are not able to bind to the antigens and stimulate the immune response again. So those two issues are what make TB troublesome. So the main site of action is in lungs, and then it can also happen in the lymph, bones, and gut. So treatment usually involves use of multiple drugs, and like I said, it must be done over a long period of time. And drugs that are usually used are rifampicin and isoniazid. I'm not sure if those are correct pronunciations. And usually, these long-term period of treatment is done by this treatment method called DOTS, which is direct observation and treatment short course. So what happens is a health worker and then the family will ensure that the patients are taking drugs and following the treatment regularly for 8 months. And it's difficult to treat TB because there might be resistance to an antibiotic. So also this helps to prevent bacteria from lingering in the body because TB can form tubercles and stay within the body for a long time. So they grow slowly and hide within the cells. So this is to prevent that. Prevention of TB involves usually administrating the vaccine, which is the Bacillus calmette-guerin. It's derived from M. bovis, and its effectiveness decreases with age. That is important that you need to take off. It usually requires more booster shots to be effective. And then also, as with most vaccines, it's difficult to keep track of vaccines along with increasing population size. And some strains of TB, as we discussed, are resistant to drugs. And over here, MDR and XDR basically means multiple drug resistant and extreme drug resistance, which means that multiple drug resistance is that TB is resistance to a bunch of drugs, and extreme means it's a lot. And usually they do contact tracing and isolation, as we discussed that in NEETS as well. Cattle usually is checked regularly and pressurizing milk to kill the bacteria. This is for M. bovis. And factors like poor housing, homelessness also make it difficult to break the transmission cycle. Usually, TB spreads in or thrives in areas where there's overcrowding, and this would be a particular issue in refugee housing because there's a lot of there's also there was a breakdown of TB programs. So partial treatment basically means there's an increase in drug resistance, and the issue with that is that only 70 to 80% who receive the vaccine are protected. So getting herd immunity is also an issue. There's antigen concealment because tubercles are forming Furthermore, there's multiple drug resistance and extreme drug resistance. So the dodge treatment require longer treatment and more expensive drugs, which make TB a very expensive disease to treat. So it might be unaffordable for those in third world countries. So these are all issues which might affect prevention of TB. So we don't really need to know much about smallpox and measles, so I'll just cover them, the basics of it. So smallpox is called for the variola virus, and it's transmitted through direct contact. And the key feature of smallpox or identifying feature is that you could see that there's red spots all over the body, and they're known as pustules. So measles is caused by the morbilli virus. So both of these diseases are caused by viruses. Um, Measles multiplies in the respiratory tract, similarly to TB, and it spreads through airborne droplet, um, resulting in a rash. So there's specific medicine for measles, and measles is also one of the most contagious. It does have a vaccine and also affects those specifically who have a vitamin A deficiency. So we've been talking about antibiotics about a lot in this chapter. So what exactly is antibiotics? So antibiotics are drugs which we take to specifically kill bacteria without harming other cells. And they have specific mechanisms on how they act on pathogens. So one of them is being inhibiting protein synthesis or inhibiting cell wall synthesis or inhibiting acid synthesis. So what this does, it prevents translation or can prevent nuclear division as well. So if it prevents translation, they cannot make essential proteins which are required for um, the pathogen. Inhibiting cell wall synthesis means that the cell wall will be so when water is taken in, might not be able to withstand the outward pressure, causing it to burst and killing the pathogen. We can rupture the plasma membrane, so it loses its essential nutrients. It can also inhibit the synthesis of metabolites. Metabolites are substances which are required for metabolic reaction. So if these substances are not made, so obviously the metabolic reactions can't take place. And also lastly, they can inhibit action of enzymes. They have no effect on virus because they don't have any specific cell structure that they can act on. Furthermore, virus can also hide within human cells or cells in general. So they may not be able to act on it because they're not able to. Penicillin is a type of antibiotic which you need to about specifically. So what it does is that it prevents synthesis of cross links between the bacterioglycan cell walls. So eukaryotes and prokaryotes, there's a difference in their cell walls, as we talked about in chapter 1, the bacterioglycan or cellular cell wall. So prokaryotes have a bacterioglycan cell wall, and penicillin prevents cross links, which are present within the bacterioglycan cell wall. They do this by inhibiting an enzyme that builds the cross walls. So the enzyme, which is known as autolysis, they make holes in the cell wall, which allow the chain to link together. Um, when it allows this to happen, it allows the cell wall to stretch. So what happens is the cell wall grows progressively weaker. And as we talked about this, so what happens is when water is taken in, it cannot withstand the pressure, eventually bursting. So what happens when, say, a pathogen has antibiotic resistance? So TB has penicillin resistance. So use of penicillin won't work on TB because it has the genetic code which codes for B-lactamase, and B-lactamase can break down the structure of penicillin. And you must be wondering why antibiotics don't work um, for example, why penicillin doesn't work on human cells is because we have different cell walls. So the features are different between prokaryotes and eukaryotes. So it depends on their mode of action and why it doesn't target human cells. So how does antibiotic resistance arise in the first place? So the first step of antibiotic resistance is mutations, and mutations are random events. So what happens is that when we expose bacteria to a sublethal dose, sublethal meaning that the dose given causes some pathogens to die, leaving some alive. So this would result in a spontaneous change in gene structure. So what happens when the gene structure changes? It may result in obtaining the sequence which codes for antibiotic resistance. And antibiotic resistance can spread by two ways, which is vertical transmission and horizontal transmission. Vertical transmission causes one species to be resistant, but horizontal transmission can cause antibiotic resistance to spread to different pathogens or different species. So Vertical transmission is when bacteria reproduce by binary fission. So the chromosome, or if the um, code which is present is on the chromosome, will be passed on to the daughter cells. So the genetic code is passed on to the species or the daughter cells. In horizontal transmission, we talk about conjugation. Conjugation occurs between different bacteria. So plasmids are often transferred between two species, and DNA may be present on the plasmid, and the antibiotic resistant gene itself might be present on the plasmid, which will be transferred through the tube. So the entire process of transferring the plasmids is known as conjugation, and obviously it can have there can be bacterium of species A, for example, and another bacterium of species B, and they themselves can have conjugation between them and spread uh, exchange their plasmids. So they can exchange antibiotic genes and stuff like that. So to prevent antibiotic resistance, what we have to do is that we ensure that patients take their full course of medication. And the reason why we do this is to avoid exposure of the sublethal dose of antibiotics. So it prevents some bacteria from surviving at the end of. You can reduce misuse of antibiotics by educating the public to take antibiotics only when necessary. So when it would be advisable to take antibiotics when it's with bacteria or and not with viruses, or not using antibiotics when you feel lucky or when you just feel under the weather. Misusing antibiotics results in greater selection pressure. So greater selection pressure results in causing the bacteria to evolve and obtain resistance. You could reduce the sale of medication over the counter. So this results in people not just taking medication when they just feel ill randomly, requiring a prescription for giving medication. You could avoid using wide spectrum drugs. Using wide spectrum drugs are obviously more useful than narrow spectrum. And if, say, bacteria start getting resistant to the wide spectrum drugs, that would be an issue. Reducing use of antibiotics in agriculture. So what farmers do is that they use antibiotics to prevent rather than cure. So spreading the antibiotics on the crops prior to an infection. So do not use it to prevent, just use it for curing possible illnesses. We could develop new antibiotics. So, and when we use one type of antibiotic for a long period of time, so there's a greater chance of bacteria evolving resistant to that one type of antibiotic. So it could do that, it could exchange it, and you could always use other ones. So prevent exposure to one type for a long period of time. Could also use multiple drugs. So there's an increasing chance of killing the pathogen at the end of the day. So there's lesser chance of any surviving at the end of the day. You could also keep testing the antibiotic effectiveness. So you know that whether the pathogens are starting to get resistance, you could start other possible use of other antibiotics to prevent a larger population of pathogens from getting or obtaining resistance. So let's just look at this diagram about vertical and horizontal transmission. So vertical transmission is when it goes from parent cell to daughter cell. So this is binary fission and or cell division of prokaryote. So bacterial chromosome and plasmids, they are transferred, and then if the resistant gene is present in the chromosome or the plasmid, it is passed over to the daughter cells. For horizontal transmission, this is mostly towards plasmids. So when plasmids are transferred, this is a process of conjugation when there's a tube. Then the plasmid is being transferred from one to the other. So this diagram is from Save My Exam. So if you found this illustration useful, you can check out their website. They have good notes. So let's just have a short summary. This is a big chapter, so I just decided to have a short summary. So cholera is spread by Vibrio cholerae, which is a bacterium, and the method of transmission is foodborne or waterborne. Malaria, Plasmodium, and it has 4 different species. It's a Protocris, which is a eukaryote, spread by insect vector, female Anopheles mosquito, blood transmission, or mother to child via placenta. HIV is caused by the human immunodeficiency virus, and this is usually through exchange of bodily fluids, for example, sexual intercourse and sharing needles. TB is caused by microbacterium and bacterium. By droplet transmission, undercooked milk or unpasteurized milk, undercooked meat or unpasteurized milk. Measles is caused by morbilli virus, which is a virus, and this is airborne droplets, coughs and sneezes. Smallpox is eradicated, so we'll talk about that in chapter 11, but it's caused by the variola virus. It is a virus, and it's usually by direct contact with other people. So let's cover some question and answers to test our knowledge on this chapter. So the diagram shows some pathogens that cause disease and some of the diseases that transmit it. So we have pathogen here, Bacterium Protoctis morbillivirus, and method of transmission contaminated mosquito bite, coughs and sneezes. So they're asking what is the correct pathogen and method of transmission for cholera. So as we just discussed, cholera is spread by Vibrio cholerae, which is a bacterium, and usually by foodborne or waterborne. So the answer should be 1. So the answer is A. Different antibiotics function in different ways. It is important that the antibiotic kills the bacteria but does not harm infected human. So the antibiotic tetracycline has been found to affect the way in which human mitochondria function. The which statements could explain why human mitochondria function is affected by tetracycline. So the options are the antibiotic prevents synthesis of peptidoglycan cell walls, prevents synthesis of linear DNA, prevents translation of circular DNA, prevents translation by binding to 70S ribosome. So obviously A is wrong because mitochondria doesn't have peptidoglycan cell walls, and it doesn't have linear DNA. It does have circular DNA, but what happens to the circular DNA? So in chapter 6, we learned about the DNA must undergo transcription, and transition occurs at ribosomes. So the option is D. So this is not exactly question from chapter 10. It's just a combination of chapter 1, chapter 6, and chapter 10. So this is one of the variants of type of questions that you need to learn to answer. So the next question. The diagram shows properties of a disease, which shows the properties that are common to tuberculosis, TB, and measles. So it's a one diagram with bacterial, infectious, and transmitted by airborne diseases. Transmitted by airborne droplets. Sorry. I'll give you a second to think about it and think about it yourself, and I'll explain. So measles is caused by a virus, while TB is caused by bacteria. So obviously option A, C, and B will be wrong because this involves bacteria. But infectious, yes, because both of them cause spread from person to person, or it's transmissible. And transmission by airborne droplets, that's absolutely correct. So the answer should be D. So common to infectious and transmitted by airborne droplets, which is D. So this next question says, why is it necessary for a person with a bacterial infection to take antibiotics and even space time intervals? And the options are to increase the concentration of antibiotics slowly to a level which is lethal to the bacteria, maintain a concentration of antibiotic which is lethal to the bacteria, prevent development of resistant strains to a bacteria or bacteria, and select and kill resistant strains of bacteria. So the answer for this would be B. And as we discussed, whenever we give antibiotics, it's usually at a lethal dose or to maintain a lethal dose because we don't want to give them or maintain a sublethal dose. So when we maintain a sublethal dose, there's a chance of survival of the bacterium. So if they survive, there's selection pressure on them. So if there's selection pressure on them, there's a chance that they will mutate. And if they mutate, they can achieve resistance. So the best answer for this would be to maintain concentration, which is lethal to the bacteria. So the answer would be B. So that's the last question for this chapter. If you have any questions, you could leave them in the comment section. All the best for your studying. I hope the videos are helpful in aiding your understanding biology. The concepts aren't too difficult, but I guess they can get challenging. So I obviously hope that these videos are helping you out. If you found them useful, you could consider subscribing or leaving a like on the video. And do definitely consider sharing them with your friends so more people can benefit. So until the next video, all the best for your studies. Have a good time.